Hey everyone, welcome back to the channel MLT Lectures. If you are new to this channel, here we discuss everything about medical laboratory technology course. So don't forget to subscribe the channel and hit the bell icon. Today we will discuss about another branch of pathology, that is histopathology. Before moving towards the video, we will first discuss about what is histology and histopathology. Here, histology means study of a normal tissue and histopathology means study of a diseased tissue. When a term pathology comes, we must always remember that it means study of a disease or abnormal tissue. So don't get confused between histology and histopathology. Histopathology is a Greek word which is sum up of three words. They are histo, patho and logy, where histo refers to tissue, patho as suffering and logy as study. So, a branch of pathology that deals with study of diseased tissue is termed as histopathology. These are some terminologies which we must know before studying histopathology chapter. These are biopsy, autopsy, autolysis, putrefaction. Biopsy means removing of a bit of tissue from a living being. Autopsy means removing of large tissue from or organ from a dead body. The difference between biopsy and autopsy is removing of a tissue from living beings is called biopsy, whereas removing of a tissue from a dead body is called autopsy. Then we have autolysis, destruction of tissue due to its own enzymes, putrefaction, destruction of a tissue due to environmental bacteria and organisms, microorganisms. Purpose of histopathology, or why histopathology is done. First point is, it is used to differentiate between normal and abnormal tissues. Second point we have is, some disease can only be diagnosed by histopathological technique. For example, bovine spongiform encephalopathy can only be diagnosed by histopathological technique. This disease is also called as mad cow disease. Now let's look in detail about histopathology process. First one is specimen accessioning. The tissue specimen in the laboratory are usually collected outside the lab, for example, operation theater room. So when it is received in the lab, should be checked properly. Samples should be labeled along with the requisition form. It must include patient demographic like name, age and sex, detail of a history, anatomic site of a biopsy, number of container sent. This is how the tissue specimen container must be labeled along with the requisition form. These two should match with each other. After a specimen is brought to the lab, the next process we do is grossing. Grossing of a specimen is an important stage in a surgical pathology. It involves accurate naked eye description of intact specimen, correct method of sectioning, cross-examination of cut surface, selection of a proper tissue blocks for microscopy. Histopathology specimens are very vital in a patient care. They not only establish tissue diagnosis but are crucial in clinical management and provide important prognostic data. Grossing is an art of knowledge what needs to be taken for microscopic study is crucial for final diagnosis. These are some general principles of grossing. It includes specimen identification, identify all the anatomical structure present, then orientation markers should be identified, then measurement is taken, length, breadth and height along with the weight is measured. After that, examine the external surface. After the examination of external surface, cut all the organs at the interval of 1 cm thickness each. Then describe the cut surface, identify a pathologic process. If suspected lesion is present, measure it and describe with color and consistency. These are the instruments used in grossing: forceps, blade, ruler, cutting board, scissor, knife. We must always wear gloves, gauze and apron to protect ourselves before grossing is done. After grossing is done, the bit of a tissue is kept into a tissue capsule and further process of tissue processing is performed. Tissue processing. It is designed to remove all the extractable water from tissue, replacing it with support medium that provides sufficient rigidity to enable sectioning of the tissue without damage or distortion. Tissue processing has various steps involved in it. Factors affecting rate of processing are agitation, heat, viscosity, pressure. Stages of tissue processing are fixation, dehydration, clearing, infiltration and embedding. First we do is fixation. The most commonly used reagent for the fixation is 10% neutral buffered formalin. Formalin is the most common used fixative. The tissue bits should be of size 2 into 2 cm and 2 to 3 micrometer in thickness. These bits are then placed in a metal cassette or capsule, which are then placed in the fixative. For tiny biopsies or small specimen, they are wrapped in the filter paper and put in the cassette and fixed. To help in visualization of small fragments of tissue during embedding, a few drops of 1% eosin is added. Fixation prevents autolysis and putrefaction of the tissues. After fixation is completed, we go for dehydration process. Dehydration means removal of free unbound water and aqueous fixative from the tissue components. Dehydrating agents are strong hydrophilic in nature. Dehydration should be accomplished slowly. If the concentration gradient is excessive, diffusion current across the cell membrane may cause cell distortion. So, we go from low concentration gradient to high concentration gradient of dehydrating agent. The various dehydrating agents are ethyl alcohol, ethanol, acetone, isopropyl alcohol, methanol, glycol. After dehydration is completed, clearing is done. Clearing agents act as an intermediary between dehydration and infiltration solution. They should be totally miscible with both the dehydrating fluid and embedding medium. Choice of clearing agent depends upon the following points. It includes rapid penetration of tissue, speedy removal of dehydrating agent, ease of removal by melted paraffin wax, minimal tissue damage, low toxicity, low flammability, cost and convenience. Some examples of clearing agents are xylene, toluene, chloroform. Next step is infiltration and embedding. The most infiltration and embedding medium is paraffin wax. It is a mixture of long-chain hydrocarbon produced by mineral oils. It permeates the tissue in liquid form and solidifies rapidly when cooled. It is compatible to most routine spatial stain as well as immunohistochemistry protocols. Some other embedding media are resin, gelatin, and celluloid. For embedding, it involves the enclosing of properly processed, correctly oriented specimen in a support medium that provides external support during microtomy. It should provide elasticity and resisting distortion during sectioning. This is how embedding is done. After embedding, a proper block is made. So, the next step is section cutting or a microtomy. Microtomy is the means by which tissue is sectioned and attached the surface for microscopic examination. The instrument used for doing section cutting is called as microtome. There are various kinds of microtome that we use in histopathology lab. This we will learn in another video, so don't forget to subscribe the channel. After a proper slide is made, the slide is then next stained with H&E stain. This stain is the most common for histopathology lab. After staining the slide, mounting is done. There are various mountants that can be used. Some examples of mountants that are used in histopathology lab are DPX, Candida balsam, colophonium resin, and terpene resin. Thank you. Hope you like the content. Don't forget to like, share, and subscribe the video.